Hello, I'm Kumba.
I'm Alison Cook.
And we're here to just give you a brief overview of what the ImGen project is all about and how you can utilize the immense data sets on our website. So the Immunological Genome Project was started in 2008, and it's a global consortium of immunology and computational biologists that seek to understand gene expression profiles in different immune cells and also to reconstruct gene regulatory circuits in these cells. So the consortium is made up of 20 labs, primarily in the US, but we also have some that are in Israel, Canada, and Australia. In the US, we have few labs at the Harvard Medical School, and we have other labs at Washington University, University of San Francisco, and some New York lab. Alison and I are from the CBDM lab, and we are based at HMS, Harvard Medical School. And our lab is mainly focused on alpha beta T cells, Tregs, FOXP3, but then as you can see, you have other labs that are B cell labs, macrophages, stromal cells, and you have the mainly computational labs, the Shea lab and the Mustafavi lab. And ImGen is currently in its third phase, primarily supported by funds from the National Institute of Allergy and Infectious Diseases, and also by Legend that generously provide most of our reagents and antibodies. And in this presentation, we'll later discuss some of the data that are available from phases 1 and 2 of the ImGen project, and also the cellular project that ImGen has been involved in.
So these are some of ImGen's major data releases that are publicly available for use in your own research projects. And then I also wanted to give you all an idea of some of the data sets that are going to be released in ImGen 3, which we're really excited about. So ImGen's most expansive data set is its bulk transcriptome data, first collected via Affymetrix microarray projects. And now we've moved to using ultra-low input RNA-seq methods, which allow even the most rare cell populations to be profiled, creating robust data sets that we release on Geo and also on our own website. So one of the great strengths of ImGen data is that we have expert labs prepare samples in a super standardized way so that data across many different immune cell populations is comparable with either data generated in your own lab or across ImGen's consortium. So right now we have a brand new RNA-seq expression skyline, which Kumba will demo in just a few minutes, with over 100 key immune cell populations. We just launched this, we're really excited about it, and it's a great resource. More populations are going to be added to this browser in the near future, so stay tuned for that. And then last year, we launched ImGen's first ever open source project with a call for applications in Nature Immunology for labs that study mononuclear phagocytes to suggest cell populations they wanted to profile and they thought would be valuable for broader consumption. So that data set was generated by over 18 different labs at institutions across the globe. We have collected this data and are processing it for release in the very near future. So if you're interested in monocytes, macrophages, dendritic cells, or microglia, this data set will probably be very valuable for your own projects, and we hope you check it out soon. Another really large data set ImGen made available in phase 2 is a chromatin data set generated using the ATAC-seq method, again with over 100 key immune cell populations, including more rare populations like red pulp macrophages or mast cells. So we hope you check that out. We have a browser called the Chromatin Explorer, which we'll also demo, and we're going to generate more chromatin data to supplement those immune cell populations, and also looping data with genomic organization and structure using Hi-C and Hi-CHIP during ImGen phase 3. So we're gearing up to do that now. And last but not least, we hope to have single-cell transcriptome data for all of the cell types that we're doing bulk RNA-seq on. So you're able to compare variability from cell to cell within a population or even across cell populations. And we think this could be really valuable, especially in challenge models or disease states, looking at how different immune cell populations react to infection. So definitely check that out in the coming year. We're hoping to release preliminary data on that in the next few months. So now we're just going to go through some of the ImGen website applications that we hope will be helpful and of use to your own research projects, and to give you an idea of some of the resources that are available on the ImGen website. I'm going to switch to a screen share so you can follow along with us.
Okay, so this is the ImGen website. It's just imgen.org, and we are going to demonstrate a few of the applications that we have, as Alison mentioned. So let's go to the gene skyline. This was generated by both RNA-seq data and microarray data. So this is the RNA-seq skyline. Let's say you're interested in understanding the expression profile of a particular gene in different immune cells. So you will use this. And let's say we're interested in knowing how TLR4, which is our favorite gene, is expressed in different populations. So here you can select your population of interest if you're mainly interested in B cells or dendritic cells. But for now, we are going to do all. And for local just give you a precise view in a particular cell type. But then for here, since we're doing all, we're just going to do global. And these data are normalized by DSeq2 from Bioconductor. And the expression values are mainly for all the population replicates that we received, because usually we do 2 or 3 replicates per a particular population. And if you scroll over, so as you can see here, since it's global, it's mainly neutrophils in the peritoneal cavity. But if we do local, then you can actually see how the expression of TLR4 varies from different cell types. So if you put the cursor over the cell population, then it can give you the long name of the cell, the population that you're looking at, because sometimes the nomenclature can be confusing, but you can see that. And also there are fax plots available for you to sort these cells in your own lab. And let's say, for example, you actually want the values, and this gives the value in a tabular form. And here you can see the different cell population and the expression value for TLR4. Here where it's 0.1, it's mainly because in this particular cell, TLR4 was not expressed, but instead, for normalization purposes, instead of using 0, we use 0.1. So this is very informative if you want to look at different expression in different conditions and also different stages in the immune cell development. So the next application that we are going to look at is the Chromatin Explorer. And as Ali mentioned, this was generated by the ATAC-seq dataset, and over 100 cell populations were profiled. So this mainly tells you about chromatin accessibility of a particular gene neighborhood, and it gives you a detailed information on the significant peaks in that particular region. So let's say we do TLR4 again. And if you know where the enhancer for this particular gene is, whether it's 10 kilobase upstream, you can adjust this value. You can do, as you can see here, and this takes you to the UCSC genome browser that you may be familiar with, that gives you the visualization of the different peaks that are available. And also for our website particularly, you can download the data, and it gives you again in a tabular form, and this is how it looks. And this gives you the different peaks, where they start, and also different predicted transcription factor motifs that are around that gene neighborhood. And it gives you the FIMO score and the p-value of those particular peaks.
So really quickly, I'll just point out two comparison tools that are frequently used on the ImGen website. One being the population comparison tool, which allows you to drag and drop specific cell populations you're interested in and receive the fold change values between the two different populations you selected. So just click submit, and in about 5 seconds you'll receive fold change values of gene expression between the two populations you dragged and dropped up here. And it even will give you p-values of statistical significance. This is really useful if you want to find out which genes are significantly upregulated or downregulated in your population of interest. And lastly, we have this feature called My Gene Set, which allows you to import your own list of genes that you're interested in. Here's a sample one I prepared. You can just drag and drop your list of genes, and let's say I want to look at these genes in B and T cells. You can create 3 different types of plot. W plot, where each point is a gene on your list, and you can see that there are some genes highly upregulated in B cells relative to T cells in the list I dropped. And then also heat map and an interactive heat map showing the same data. And this is a really quick and easy way to make figures for presentation. If you are interested in gene expression in different immune cell populations we have available on the ImGen website. So we encourage you to play around with those tools, and feel free to email us at imgen@hms.harvard.edu with any questions, or if you want help finding a specific data set of interest to you. We really hope you use this as a resource in your own projects. So I think now we'll take some questions, and feel free to reach out to us. Thanks.
Useful and informative tool. I'm just going to go through a few of the questions asked from our global audience. So we have one asking, what is the deep RNA-seq function used for specifically, and can you comment on it? And if you have time, maybe demo it very quickly.
Yeah, the tool on the ImGen website, you mean?
Yes.
Yeah, let's see. I can screen share again and I can just pull that up if we have time.
Yes, we have a few minutes, so please go ahead. Show us your expertise.
Deep RNA-seq. Okay, so another cool thing to note is that all of these features have this little question mark, which gives you more information on the tool itself. So the Deep RNA-seq feature shows you the UCSC traces for deeper RNA sequencing we've done with a really high number of reads, and allows you to zoom around on this visualizer specific genes you're interested in across all of the populations we have on file. And also you can use a lot of the data tracks that are already available on the UCSC genome browser in tandem with ImGen's own data. So if you have your own track you want to add to this, or if there's specific alternate splicing or additional information you want to overlay with our data set, this is a really quick and easy way to leverage that.
Very, very cool. We also have a couple general questions. Is there data available through ImGen on where the samples came from? So for example, which group, male or female, which age distribution, etc.
Yes, definitely. So as I was sharing in the gene skyline, if you go to the microarray, and when you put the cursor over the gene name, it tells you the detailed description of the sample, so where it came from, the lab that prepared the sample, and also detailed description on the sorting marker, the age, the gender, and also the different conditions. So whether it was injected with a particular bacteria, or whether it was done by plating or replating. So all those different information are there that you can see.
Okay, and in terms of using the interface, what type of gene notation does the ImGen website accept? You are importing your own set of genes.
If you're importing your own set of genes for the My Gene Set, we just use the standard NCBI gene names. You can Google with your own annotation and it should come up with the standard NIH set.
Okay, we do have one or two more questions in the chat box, if you guys can take a few minutes to address them.
Sure.
Actually, we'll take one. So one question from Sergey at U of T Toronto: is it possible to perform convolution using ImGen? So for example, if you had data from human whole blood.
I'm not sure this is a feature we have currently, but it's something we're hoping to introduce with the single cell, where you could look at whole tissue samples and then tease apart different immune cell populations from those. But definitely a cool suggestion and something we hope to introduce.
Okay, thank you very much, guys, once again. I do anticipate you will be receiving a ton of questions as people discover the utility of this tool. So just if you can even provide your contact information that you're comfortable sharing with the audience in the chat box, that would be amazing. Thank you once again.
Great. Thank you for having me.